Hello. Okay, I guess so far the first experimentalist. Um, so the talk there won't be any. So your cognitive load will be low today in my talk because there's basically no math. So the only equation I will show is the purpose is to show it's probably wrong and need to be updated. So um, so how many of you are? I just still want to know how many of you are experimentalists or experiment? There are, but how many of you for the experimentalist? How many of you have done electrophysiology? Patch clamp. Okay, that's good. Okay. Yeah. But still, a great proportion is the doing modeling or theory. It's not working. So, okay. Yeah. Okay. Just recap from Rava the other day. So the title is why for you guys to need to talk to each other, the theorists and mentalists need to talk to each other because first we have a common goal as Rava says, to understand how the brain works, right? And then you need to analyze experimental data, so you have to talk to experimentalists, get data from them, okay? And then you need to quantify the behavior in animals, so animals and neurons. So those are really from experiments, right? You have to find someone can train mouse, can track zebrafish, and then you need to identify relevant facts and extract complexity away, right? That's from Rava. Then what's the facts? So there are so many facts, there are so many neurons, so many processes, dendrites, axons, molecules. Which facts are relevant that need to be incorporated into your theory, into your model? So you need to talk to them. Okay. So then you need to propose in the end, if you have a very nice theory you want to propose, then you need to let the experimenter know. They need to have intuitive understanding of your theory. Then they can do experiments to test your idea. Otherwise, it's the value will be not that high. So okay, so that's why we have to talk to each other, and you probably that's why my talk is important. I will tell you a lot of facts, and the facts maybe you think you know, but I will give you a fresh angle. Okay, the same pictures Rava shows. This is really the challenging part. The brain is so complex in many different levels. There are so many numbers, huge number of neurons, huge number of synapses, and those single terminal vesicle release, action potential comes in, how the single action potential can drive vesicle to communicate with downstream neurons, and then how different neurons converge, and then how different brain area interconnect to give rise of different level of functions. This is so challenging and even frustrating. So what we do, so basically, there are currently two type of approach. Okay, I think I rarely find people talk about this, but I found Chuan, one of the Chuan's talk about this. So there are basically two type of approach. One is bottom-up approach. Essentially, this is taken by the Blue Brain Project in Europe. Henry Markram, people like that. They propose they need just to reverse engineer the whole brain or neocortex. To do that, you just, regardless of whatever function from bottom up, which is measure everything, measure neuronal morphology, measure connectivity, measure firing pattern, measure synaptic physiology, After measuring, then we can digitally reconstruct all these parameters or facts. And then somehow, hopefully one day they can use supercomputer to simulate. Okay. Then the last hope, the bottom-up approach, people always have this last hope of emergence. Okay. Somehow, some kind of interesting function maybe one day can emerge. That's emergent property. They think, okay, maybe consciousness can arise, magically emerge from the population of simulation. Okay, so this is a bottom-up approach, but turn out it's too frustrating, too costly, and you probably will lost in forest. You have so much facts. You have probably so that hope is probably very small to have something emerging. And then there is a very smart, brilliant idea from David Mars many years ago. This is top-down approach. Okay, so there's he proposed three levels of understanding. First is computational theory. Essentially, needs to define the computational problem that the brain needs to solve, whether it's vision, whether it's color vision, whether it's decision making, whether it's motor region. So you need to define the problem, and with a very clear theory, or at least you know what the brain needs to compute. And then you need to see, oh, okay, what kind of algorithm the animal is using. This is very important because the same function can be implemented or realized by many different algorithms. Essentially, the same function can be realized by LLM. Okay, they use a very different algorithm than the brain. Then some tasks they can do it very, very well, much better than us. Okay, so there could be many types of algorithms. Then that's the problem. If we don't know the algorithm, how can we study the implementation? So the final level is the hardware implementation. We need to know how the brain, the neurons, the biological materials, tissues, how those things can implement those algorithms. So then we need to really know which algorithm the animal is using. So that's the difficult part. So the top-down approach also has a challenge. So you probably were stuck in thinking, what algorithm is the animal using, and what computational problem I need to study, and then how the brain implement. So you really don't know what to do. Okay, so that's the thing. So one way is how about we combine those two approach? Okay. Yes, we combine this in a smart way. Maybe we can have some hope. Okay. So for example, starting from bottom up, we measure a lot of things. Okay. Once we know something about the facts, we can constrain those algorithms. So maybe some algorithm is possible, some are not. For example, Mitya talked about this local learning rule. So the backpropagating global backpropagating is not possible. It's not biologically possible. So then at least we know something. So then some kind of algorithm is not the brain using. So there are we study a lot of details. So there are a lot of experiments. They just start without any hypothesis, data driven, just measure things. But then their measurement, you can try to know, to learn, and then use those facts to constrain your idea. Then you maybe know, okay, if there's one algorithm is working, maybe you can try to do some kind of experiments. How different cells connecting to each other, one cell fire, the other cell is inhibited. So how neural circuit can implement some of the algorithms. And then there are you need to define the problem. That you cannot inspire from here. So you need to study behavior. So in that case, you start from top-down approach. You define a behavior, train the mouse, and study the human. Highly well-controlled, in well-controlled behavior, characterize everything. Then you define the problem. Then back and forth. So probably there will be hope to do something. Okay, so So today, the first talk, okay, mainly talk about from this approach. Hopefully reach some level of this. And the next talk, I'll starting from this and go down. Okay. So that's lecture one. I will give you a lot of background knowledge. What's the structure of neuron and cortical circuits, and what's the functional property of the neurons and the circuits. Maybe you read a lot of, or already learned a lot from textbook, but most of them are wrong. I'll give you probably hopefully, okay, there's something new you can learn from today. And then because many of you are doing theoretical work, so I'll introduce some kind of basic concepts of how to measure neural activity. So it's important for, if you do modeling, it's important to know how the data are measured. Okay, the measuring approach should at least have some concept, have some intuition. Okay, and then the final one is giving you some our study to show how we are using this type of kind of bottom-up approach to study the neuronal circuit mechanism or processing during behavior. Okay, the first structure, morphology. Okay, I guess you know neurons are not a regular cell. They have they are different from other tissue. They have this dendrites and axons. So how many of you really know dendrites and axons? That few? Okay. It seems after I talk, most of people are losing confidence. Maybe I don't know dendrites. I don't know axons. Okay. I think most people don't know. Because Wikipedia, if you look at Wikipedia, this is how neuron look like. When you look at this type of neuron, it's only a few, stub, branch and short axon, a huge cell body. What do you think? How can you imagine these dendrites are doing anything interesting? How can you imagine this single axon sending out doing any complex thing? It's hard to get from this kind of popular image. This is really from Wikipedia. So this is teaching people how neurons look like, and this image is very misleading. Okay. Then I'll show you something most of the theorists and model maybe you don't like. So and most of the model are using neuron as point source. There's no axon, no dendrites. They just connect with each other. Even the famous Hilbo and Whistle model, there are points. They're converging feed forward, and then visual perception appear immediately, magically by feed forward. That's wrong. Okay, and then third bottom. Okay, but the beautiful dendrites. The beautiful dendrites of Purkinje cell is now becoming this, and there's no dendrites, right? And Hopfield model, okay, no dendrites, no axons. Yeah, the axon is just ignore the length, ignore the complexity, just one line connection. And this most popular model integrating fire, they just converge and sum and fire some kind of fire rate. I think this really will upset our founding father. This is how modern neuroscience was born. His founding father, the most amazing work is he can draw those beautiful structures, a lot of details. This is beautiful Purkinje cell and beautiful pyramidal neuron, leaf 5 pyramidal neuron. The most prominent part of that, the dendrites, It's a huge like tree-like structure. So when you look at this neuron, you will start to wonder what the dendrites is doing. But if you look at the Wikipedia, oh, the dendrites is boring. It can't be doing anything interesting. But here, why are they standing such long apical dendrite? 2 layer 1 and branching out a lot. Why they branch out so much? You know, if you are doing science, you need curiosity. You need to ask questions. Why are they branching out so much? Are they receiving very important information there? What is the information? Why they receive those information? Why they're so far from soma? If those information are very important, but they are very far from soma, how can those information impact the cell output? And these very curious questions. You know, if you use a point source, if you look at Wikipedia, you cannot answer this question. You won't be curious about those questions. And the same time opponents of Cahar is this Goji. Well, his theory is reticular theory. His drawing is okay, no more complex dendrites and spines. Those spines. He think those are just artifacts. You know, Cahar used his method of silver staining. Golgi staining. So you think those dots, those are just artifacts in my method. But turn out those are very important synaptic connections. The location of synaptic contact, the spines. Okay. So really, the boring of modern neuroscience is based on those drawings by Cahar proposing the neuron doctrine. The neuron doctrine is not just a point neuron, remember? Okay. The neuron doctrine are these diverse and beautiful structures. Okay, so these are dendrites. But Cahar, in his time, because he can only slice locally, but he cannot reconstruct the entire axon. Okay, so he does not have this fancy microscope. But now we have. So one example of the axon is this colostrum neuron. Actually, that neuron is proposed by Francis Crick and Christoph Koch as probably the conscious neuron, because they really innovate almost the entire brain with their axons. Okay, so this is the modern time imaging. We can reconstruct the axons of a single neuron by slicing and imaging the entire brain of mouse. And then this is one example of complexity of axons. So axon projecting to everywhere, to multiple brain area, a long distance. And then you can ask, okay, if a single action potential travel such a long distance, reaching so many terminals, are they sending the same message? Are they effective in communicating by single pulse? Those are curious problems. If you know a little bit more about detailed experiments, the synaptic transmission mechanism, the vascular release. The vascular release, there's a lot of machinery to regulate. And there's like readily releasable pool, and there are like secondary pool. And then the one spike goes there, may fail to trigger any vascular release, or may just release negligible number of vesicles. And then there's this type of regulation will allow for the neuron to use a different type of communication. For example, a train of action potentials, a burst. If you're sending a burst, then the synapse will be very reliably released and communicate with downstream. A single action potential often fail. I'll propose something about single action potential, their role. All right, so today we have a new study, a new large scale work to reconstruct the 6,000 prefrontal cortical neuron, their entire brain with single axon resolution So these are data sets available, and these are recently published in Science reconstruction of 10,000 hippocampal neuron projection pattern of the entire mouse brain. It's also available. So for theorists, the modeler, those are information that may be useful if you want to check, if you want to know, okay, when some type of cell, I want to know which brain area they want to connect. You can go to the website and look into the database and find, okay, your answer. May be helpful for your model, for your theory. Okay, so that's about axon. Okay, now you know dendrites, axon. You know that's the structure of a single neuron. Okay, I hope you have something new, learned something new. And finally, as I said, there is synaptic connection. Okay, the synaptic vesicle release. So it's also important for modelers and theorists to know some of the detail how the vesicle is regulated and those post-synaptic receptors, how they can be activated, how under what condition NMDA can be NMDA receptor can be activated, so that calcium can influx and produce either calcium spike, either NMDA spike or calcium spike or synaptic plasticity. Okay. All right. So that's basic knowledge, and then there is basic functional properties. So membrane potentials, EPSP, and action potentials. I think everybody knows. And circuit level, feed forward, feedback, and local inhibition. So I wouldn't have time to talk too much about these, but I'll briefly mention the membrane properties, which are actually important if you consider the rich morphology. So the passive membrane property means the capacitance and the resistance of membranes. Because of this passive property, the rich structure of the dendrites can have a very a big impact on their electrical signals. For example, for in spine, in single spine, because the local impedance is very high, as long as there are some kind of channel open, ion influx, the same current can cause a huge depolarization and fast because the membrane area is small. So the signal there can be large and fast, but the signal in the soma can be small and slow. So different type of depolarization can activate different type of ion channels and leading to different type of nonlinearity. So those details are important. That's why people are doing realistic modeling, multi-compartmentalized model. They need to measure a lot of details of the membrane properties. Okay, and action potentials. Okay, so this is from the textbook action potential. Action potential is mediated by sodium channel, right? So I think everybody know action potential is all or none events. How many of you know this is regenerative events? Anyone know what regenerative means? No? Is regenerative means that comes again? Repeats. Okay. Why? Repeats? By repeats, you mean the action potential will fire more repetitively? Yeah, partially, yes. But regenerative events is actually for the same events. Yes, but not exactly. So yeah. Regenerative means it's a positive feedback. basically, why is or none and fast events? Because here you have depolarization, initial depolarization to reach threshold that opens some sodium channels, but those channels have two properties. One is voltage dependent. Two, it's electrogenesis. Means if it's open, it will change the membrane potential further. So it will depolarize the membrane potential further and activate more sodium channels. And more sodium channel will do the same thing again. So within a very, very short time, it will generate or non-events. So regenerative property will define a spike. Okay, if you say something is a spike, it should be regenerative. So then the spike action potentials is not sodium action potential. Is only one type, one of them. Any other channels, if they show this property, they can produce spike. So this is important because we have calcium spike in the dendrites. In the dendrites, a sodium spike are mainly in the axon initiation zone. There are high density of sodium channels, but in the dendrites, there are a lot of calcium spike. Especially there are high threshold, high conductance calciums, actually calcium channels, voltage gated calcium channels. Those channels also have the threshold. If you reach threshold, a regenerative events will be produced, then a calcium spike will be produced. So what's the difference between calcium spike and sodium spike? The sodium spike, sodium channel inactivate quickly. That's why it's so short. But calcium spike, calcium channel inactivate slowly. So calcium spike will be long and large. Okay, those long, slow, and large spike is still called spike, even though it's like a plateau. It's called spike. Can have very strong impact in the output. So if you continuously depolarize with huge amplitude, that will generate a burst. Okay, the burst is a very powerful output, and plus this will cause a lot of calcium influx, will cause a lot of cellular signal transduction, leading to certain type of plasticity. Okay, so action potential, not just the sodium action potential. There are calcium, so action potential, calcium spike, and I don't have time to talk. There are also NMDA receptor spike. So NMDA is, yeah, this is one way to test whether this this component is mediated by calcium channel, because this is a broad calcium channel blocker. So if you block that, these large slow events disappear. So those small things are mainly this backpropagating action potential. So action potential. So I wanted to mention this, but here I can mention this too. Action potential cannot just propagate along axon to downstream. Another very, very important role is they can propagate back to the dendrites. So this is very important. This is probably more important than their role in sending out. I'll talk about this. So those action potential will propagate back. And if you record patch clamping, if you record on the dendrites, you will see this this remain depolarization due to this backpropagation. Yeah, if in some case it will reach threshold to evoke calcium spike, and if you block calcium channel, then you only left this depolarization due to backpropagating action potentials. Okay. All right. So this is an active property. This is a functional property of neurons. I hope you learned something new. Yes. So when I talk about backpropagating action potential, is referring to sodium action potential. So sodium action potential, because normally people think it's forward propagating to downstream along the axon to communicate with downstream neurons. But at the same time, if there's a single action potential generated, it can propagate along the dendrites. This is backward. Okay. Calcium spike means if you already generate here, it can of course spread broadly, but you cannot say whether it's forward or backward because it's odd directions. Okay. Okay. So the back propagating action potential is not the back propagation in AI learning rule. Okay, that's different. But you can think about this as some kind of feedback. Within a single cell, there is feedback. This concept is very important. At single cell level, there is feedback. Okay. So yeah. Good question. I will talk about this later. Yeah. So this is a very important topic today. Okay. So functional property of neurons. I hope I give you a different view of what you normally can read from textbook, but they don't emphasize a lot of things. Okay, for experimenter and for especially for theorists, it's a very background in physics. It's probably good to mention the method how to record the neural activity because it's a class. Okay, so of course the first one I should mention is patch clamping. Patch clamping is invented by Erwin Nair and Bert Sakman. So they got Nobel Prize because of using this method to measure single channel currents. And today this method is very useful in measuring intracellular signals. Okay, this is probably the best way, the gold standard, the most high signal to noise ratio measurement of intracellular signal, electrical signal. So the way so anyone I guess very few of you have done patch clamping. I just briefly tell you how it's done. So you have a electrical pipette. You have a pipette pulling by heating, and then the tip will be sharp. The opening will be very small. The range is like 1 to 3 or 5 micron, and then the glass needs to be very clean. Then you can fill in with solutions, conductive solutions, and then with this pipette, you can approach a cell in like a brain size. Okay, here you can watch. So here is the typical pyramidal neuron in brain slice under DIC microscope. So with the pipette approaching, if you have very clean pipette, then you can make a suction, and then you can suck a piece of membrane into the pipette. Okay, in that case, if your pipette is clean, then there will be high impedance seal between the membrane and your glass wall. And then after this, if you measure impedance by delivering a voltage pulse, then you will see a very high impedance. Then next step, you just break in by a very brief suction. So everything is done by mouth. You first slowly use your mouth to suck a piece of membrane into the pipette, then do a quick burst and then break the membrane. And then you can measure whatever inside is going on. So this is patch clamping. Why this is useful? Because a series of experiments derived from Sakman lab and postdocs from there, they can measure in brain size not only from soma but also from dendrites. Okay, so that's why people can study what's the difference of the signal between dendrites and soma. So the most obvious one is if you deliver some kind of pulse or waveform into the dendrites, it will decay much strongly when you measure in soma. Okay, the signal essentially will decay. So this is dendritic filtering. So that means all the synaptic inputs arriving in the distal dendrites will decay to a very small amplitude. So this is also intriguing. Okay, and of course you can measure that with the so that's how this signal was measured by patch clamping. Okay, so these are patch clamping for intracellular recording, and then these days, yes, yeah. All right, yeah. Yeah, yeah. Oh yes. So this is just one experiment. They want to see how dendritic filtering will affect the waveform. So for example, they want to ask whether it's high pass filter or low pass filter. How that will impact amplitude and frequency basically. So it's not slowing down too much, I guess. But people consider this as more like a low pass filter, and mainly it will reduce the amplitude. Okay, yeah. But a different type of cell can have very different properties. So these are just one type of cortical layer 5 pyramidal neuron. Yeah, you know, in other brain region, other cell type, this property can be very different. Okay, extracellular recording. So I guess most of you may know because this is a very popular. People do a lot of theoretical work based on spiking and systems neuroscience. Essentially, take an electrode into the brain, a monkey brain, and measure spikes. Okay, so these are very popular. So basically, this what you can measure is use metal electrodes. If you are close by to some neurons, you'll find this fluctuation with some spikes. So these are different components. If you do low pass filter, you can get local field potential. If you do high pass filter, you can remove the slow fluctuation and you have spikes remaining. So these are all extracellular signals. Basically, when the cell are firing action potential, they can cause the electric field change nearby. And if the group of cells they are synchronized, then their even subthreshold activity can produce a large enough field change. You can still measure it. So local field potential may sometimes reflect synchronized oscillatory activity, but still it's debating like a previous talk mentioned. Yeah, so there are different type of extracellular recording electrodes. So these are classical tungsten electrodes. Essentially, you have a very sharp tungsten tip insulated by glass in the rest of the net, and that's classical one. But using this type of electrodes, people made a very important discovery, like Hubble whistle using this type of electrodes in the cat visual cortex. They did discover this orientation tuning neurons. Okay, so this is just a briefly mentioned. And there are multi-channel electrodes. So the most widely used in the earlier time is this tetrodes. There are 4 wires wind together, and if you do a cutting and coating, you will have a good conductance. In the tip, you have 4 points. The 4 points has different distance from the neuron, so that you can do some kind of PCA analysis to isolate single cell by studying the signal from the 4 different channels. So essentially with this, people construct this type of micro drive. This can be implanted on the rodent brain head. Then you slowly insert those wires into deeper brain area like hippocampus. over days, you every day you turn a little bit, and over days you can reach your target, and then you can measure with quite a good signal to noise ratio, and you can get a number of cells. So this is a classical method that was used to make this very important discovery of plate cell and grid cell. Okay. So you may notice all these are really bottom up. This is really data driven. They just insert electrode, just watch what happens, and wow, it's there's some kind of regularity. Okay, so these are actual cell recording. Then these days, it's further developed with into this high density extracellular electrodes. One of the most popular one is these neuropixels. So this is really the electrodes can give you thousands of neurons data, and these days, these type of data are the main target for theoretical and the computational people to analyze. So I will not go to any details. Okay, with this, you can basically insert the electrodes into many different brain area when the mouse is doing certain type of task. So in this case, there's a visual discrimination task. So the animal uses the wheel to turn left or right, and to discriminate the contrast of the orientation gratings, and you can measure activity from many brain areas. So so advantage of this is you really can reach a lot of deep brain area, but the disadvantage is you don't know cell types basically. So and you don't know layers, and you don't know whether you're recording from single spikes or burst, or whether there's calcium spike. It's not known. Okay, so these are electrical recording, and another main method to record neural activity is two-photon imaging. So you guys know about two-photon imaging? Why? How it's done? What's the? Okay, I'll just very quickly go through. So why two-photon imaging? What's the difference between two-photon imaging and fluorescence imaging? So it's actually very simple. So conventional fluorescence imaging is done by delivering high energy light to fluorescence molecules. So if a molecule absorb certain amount of energy, will be excited to an excited state. So that's not stable. Then quickly the molecule will jamming back to the original ground state. Then at the same time, the energy will be released, and part of the energy will becoming heat will be lost. Then a lower amount of energy will become the photon with the higher wavelength. Okay, lower energy. Yeah. So in that case, you use shorter wavelengths like blue light. We excite the molecule, and you can collect visible light of green photons. So this is single photon imaging. But people start to think if we use a longer wavelength, the longer wavelength is the more advantage in biological tissues. But the longer wavelength has lower energy. So how can a lower energy excite a molecule and get higher energy output? So but earlier physics suggests if in theory a single molecule receives the two photons at the same time, then you will have enough energy. But under natural condition, this probability is extremely low. It's basically it's not happening. However, after a long time after that theory, the industry developed this ultra fast pulsed laser. It's called femtosecond laser. So laser can deliver high energy light. But then this type of laser can concentrate all the photons within a very short pulse, within like 100 femtoseconds, and repeat that with like 80 megahertz. Okay, so then repeated deliver a very narrow pulse. In that case, a lot of photon will be concentrated within a very, very short time. Then those photons hitting the molecule, the chance of having those molecules receiving two photons at the same time will be significantly increased. And then at the same time, if you use the lens to focus that laser to a single point, then that focal point will have enough, there will be a sufficient chance to get to two photon excitation. Okay, so this is how two photon imaging is done. So the good thing is naturally it has very high spatial resolution because only at the focal point you can get fluorescent signal. There will be sufficient two photon absorption. Okay, so that's two photon excitation. Okay, I think it's easy, right? It's quite simple. And then with only single point, you cannot produce an image, right? So then you need scanning. Basically, how two photon imaging is done is you're scanning a single excitation point fast, quickly through your sample image plane. Actually, it's collecting at any time. You only collecting data from single point. But the scanning is so fast. The line scan plus the Y scan. There's a two scanning mirror working together will generate an image basically. But that image is a one frame, but it's actually across time. The first pixel to the last pixel, there's a time delay. So if you see a lot of imaging data, you should know there's a time delay for each frame. But normally, if yes, yeah, yes. It's 100 femtoseconds, and the repetition is yeah. So you're actually right. The best temporal resolution is this interpulse interval. So the sampling rate cannot exceed the pulse repetition rate. Yeah, it's 10 nanoseconds. Yeah. So it's 80 megahertz normally. Yeah, yeah. The scanning rate is much slower. So normally, so for today's resident gobble scanning, it's about 8 kilohertz. So it's much slower than this. So in that case, it's okay. But when you use a slower laser, for example, these days if you heard about 3 photon imaging, if you do 3 photon imaging, then you need to have the molecule to receive 3 photon. Then you need to further reduce the repetition rate to pulse the energy high in each pulse. In that case, the repetition rate will be much lower. Then you will have to consider whether this is sufficient for you to sample. So you have to calculate what's the frame rate you can get. So there will be limitation of the fastest frame rate you can get. Yeah. Good question. Okay, so basically you produce the two-photon image by scanning and reconstruction. Okay, so you scan the field, and then the computer knows that every pixel, what time, what space you have that value. Then you can assign that value back to that location. Then you can reproduce one image. Okay, so the good thing is with this is for in biological tissue in brain, you have very nice spatial resolution, and so that's why you can use this method to really image dendritic spines, see how the morphology change over time. For example, if there's a learning happen, then the spine where the synaptic connection is, then that the spine size can change, suggesting the connection can be changed over time. So this is you can do structural imaging, and also of course you do functional imaging. The functional imaging mainly using calcium. So so what you measure with calcium is mainly coming from different source, but in neurons there are two major sources. One is the voltage-gated calcium channel. So whenever the neuron fire action potential, the depolarize to the membrane to quite a bit, then that will open calcium channel. And then there's a NMDA receptor, which if glutamate bind this NMDA receptor at the same time, if the cell is depolarized, then there will be NMDA receptor opening that has a very large calcium conductance, and a huge amount of calcium will influx. Okay, so that's a basic concept of calcium imaging, calcium signal. Then what you can do is, with two-photon imaging, you can measure a population of neurons at the same time. So this is a very early time. It's already, you can study a population. These days, you have a large field of view and the faster scanning. You can basically acquire almost more than 1,000 neurons in each frame. And also you can study very finer structure, the calcium signal. For example, you can measure within for the same cell, you can measure different branch, how they responding to visual input. Okay. All right, so these are methods to record neural signal. Okay, until 12:30. Okay, good. All right, so after those introductory thing, we can talk about some science now. Okay. Okay, the first topic will be information processing in single neurons, dendritic integration. So one of the classical study measuring single neuron information processing was done by people trained from Burt Sackman's lab. So Matthew Luckham and Julius Zhu, they were postdocs with Burt Sackman. And this is one of the classical study. So what's interesting about these results? Let me explain. So this is a layer 5 pyramidal neuron in biral cortex in slice. Okay, what their superpower is, they can record from the same different location of the same cell. Okay, soma and apical dendrites and distal apical dendrites. Okay, and then what they want to test is because I guess Matthew has this curious question: why there is a layer 1, right? What when they receive input, how that input can impact the cell output? Okay, so so what they show is if you inject current to simulate to mimic a local EPSP. Okay, so this is the red trace. You have a current injection, and you can measure locally. You have a quite decent depolarization, EPSP like depolarization. Okay. But then that signal, if you measure from here, the blue electrode, the blue trace, it's basically flat, 0. Okay, so the distal depolarization by itself does not produce any impact of the somatic output. Okay, and then that's curious because it's why there are so many branches, and people think layer 1 there are tons of axons traveling. Why what they're doing? Okay, but then on the other hand, if you record from here and inject the pulse of depolarization, then you can evoke a single action potential, sodium action potential. Okay, I'm glad I mentioned calcium potential, sodium potential, so now I can talk. So this is sodium action potential, the blue trace, and then if you record from there and there, you will see the signal is propagating back. You will see a quite significant depolarization. So the red trace reflecting the the backpropagation of the sodium action potential. Okay. So that's the action potential only. So that's the distal input only. But if you evoke the two events at the same time, okay, then interesting happens. So the single action potential propagating back interact with the local input. Somehow, magically, a large plateau potential was generated. Okay, so this is the dendritic calcium spike. Okay, so dendritic calcium spike is slow and large, so it's why it propagate. It can the depolarization amplitude is sufficient for it to impact the somatic output to generate action potentials, and then the second one, the third one, will kick off. So essentially, the coincidence of the two events, this and this, will generate calcium spike and somatic burst output. Okay. So also in other system, it's also a very classical study by Jeff McGee and Dan Johnston in hippocampal brain size. So there are also another lineage of people who can do dendritic patch clamping. So they mainly patch clamp from hippocampus neurons. So here they show is if you pair the distal input with the somatic action potentials, if that pair was repeated multiple times, that can produce a long-term potentiation. So the reason they think they can produce long-term potentiation is also this nonlinear integration. So so here are different traces measured at this location with calcium imaging. So that's calcium signal. Okay, so the EPSP only. So if you stimulate here with extracellular electrodes to produce EPSP, then the signal is very small, even around here. And then the backpropagating action potential will produce this kind of calcium signal. And the pairing of the two events will be much larger. Okay, so that's actually significantly larger than a simple linear summation. So that means it's a superlinear integration. The same thing can be also found here if you produce multiple burst pulse. If the synaptic input is paired with the somatic action potential burst, then here one is the trace showing this paired case. So the calcium signal will be much larger for pairing. So that actually will generate long-term potentiation of the synaptic input. Okay. Okay. Together, this kind of integration will get you large calcium spike, which first it will be a very powerful output. Yes. Yeah. Yeah. Here is basically the cause. So what so with a single pairing, you cannot produce, normally you cannot produce synaptic potentiation. But here they mimic theta burst in the hippocampus. They have this theta frequency, and then each pulse there are high frequency action potentials. So this complex theta burst, there's a pairing, the theta burst of action potential plus the distal extracellular stimulation producing EPSP. The combination will enhance the EPSP size. So that's classical long-term potentiation. Okay. Okay. So so these two, yes. Yeah. So if you only inject current here, because it's not going to involve synaptic transmission. So in that case, that's why they didn't find any potentiation because the distal input only was only stimulated by current injection. It's not directly stimulating by actual by the directly stimulating synaptic transmission. Okay, but this case will produce bursting output, and the bursting output is known. It will a lot of calcium influx that can cause very significant intracellular signal transduction. Either change the synaptic plasticity or even change the intrinsic excitability. Yes. Well, I have calcium spikes without somatic action potential. That's a very good question. So yeah, it's possible. So here, although it's demonstrated one effective way of generating this is pairing. So there are other study like one by Nelson Spurston back many years ago in hippocampal slice. They show one way of so there's another way of generating LTP is local input. If the local input is coincident enough, they can generate a local dendritic spike. Okay, that the dendritic spike, if they pair with other simultaneous input, in theory, you can produce LTP. So they showed that it's possible. So yeah, so there are in the brain there are always exceptions. So in theory, if you have sufficient synchronized input, you will probably produce local spike. So at least it's already known you can produce an NMDA spike. So that's studied by Jackie Schiller. The NMDA spike I didn't mention here. So it's a local nonlinear form of yeah. Yeah, that's a very good question. That's why when I talk about this, even those are classical, there are so many open questions. So dendritic integration in inhibitory neuron is essentially it's very little known because it's so difficult. Even patching this large apical dendrites is already an art form. For inhibitory cell, it's almost so. Intralaminar. Yeah. So that's why I showed this. So basically, these are one form of large pyramidal neuron. Yeah, and there are other type of interneurons. So it's the cortical circuits is very complex and diverse, a lot of details. So and still a lot of unknowns. Yes. Yeah. That's a good question too. So it's not completely understood, but in this case, mostly it's coming from voltage gated calcium channel because you can try to do pharmacology. You can block it with calcium channel blocker, but in some case, in theory, those calcium influx can also cause signal transduction inside the cell. So there's a ER calcium store. There are a huge amount of calcium. So if those pump or channel open, that can cause much slower and rising of calcium concentration in cell. No, there are ER in the dendrites. Yeah. Uh, yeah. That kind of experiment was already done. If you block calcium channel, so there are different type of calcium channel, low threshold, high threshold. You can use different type of blocker. You can find different type of contribution from different type of calcium channels. Sorry? B and C not in the same time? Same type? Yeah, so here C and B are done separately at different time. Yeah, oh, different shape. So yeah, so here is the basically they're trying to mimic local EPSP. So the current injection is like this exponential shape. They want to mimic a synaptic input. But for here, if you just want to evoke a potential, you just simply depolarize with the square wave. No, actually, if you have only the somatic depolarization, you have one action potential. But then here means you have same time. You have first depolarization here, this pulse, and the red trace. You also produce this local depolarization that can generate depolarization here. So this events and this backpropagating action potential, they interact, then can produce nonlinear integration, nonlinear superlinear summation, basically. Was the question somewhere? Yeah. Yeah, it's a whole class of study. I don't have time to talk about, but I briefly it's also another regenerative events because NMDA receptor is voltage dependent, right? So it has a large conductance. Anything a large amount of calcium influx will depolarize further. So the further depolarization will potentially activate more NMDA receptor. But NMDA spike require another condition is the glutamate release. But normally the glutamate release is sufficient, is over the amount. So you have sufficient glutamate nearby or binding those NMDA receptor. So those are potentially already excitable. Then the channel opens, and then we'll open other channel will depolarize and remove the magnesium from the, and then if those channel they are already bind with the glutamate, they can be activated. So this type of activating state of those channel will generate regenerative events and get R or none. Yeah. All right. Okay. So yeah. I'm sorry. Yeah. In this study, okay, LTP. No, this one did not show any LTP. Yeah, but this one that did show additional interesting thing. I didn't have time. I don't want to do a journal club of this. But another thing, but I think it's worth mentioning, is if they directly activate the distal axons with extracellular electrodes to produce EPSP, in that case, it's actually very difficult to produce calcium spike. Why is that? So another important thing in their study is there is very strong inhibition. The inhibition in the neocortex, essentially it's feed-forward inhibition, activated by extracellular stimulation, and they have to do a GABAergic blocker to produce calcium spike. So that means, although so this event is very significant, but normally it has to be inhibited. So our brain cannot produce this kind of huge events all the time. So only very significant events, behavioral events, very relevant, very impactful, can generate this kind of thing. It's highly regulated. Okay. And of course, those inputs by themselves, they don't produce output. People have also hypotheses. Okay, I'll talk about this later. Okay. Yeah. So put this back to the context. I already mentioned there are a lot of local inhibition. So there are different type of local inhibition cells. One is PV, a different class. So the thing I want to mention is the PV cell mainly targeting soma or parasomatic area, clamping the spiking. And the somatostatin interneuron often target dendrites. Will try to inhibit dendritic events. So all these events are highly controlled by the local circuit. Okay, so essentially this is a summary of the integration mechanism. So before integration, there is a single action potential, and then if the single action potential backpropagating at the same time, there is a feedback input in the distal dendrites that can generate a large spike and produce bursting output. So this actually suggests, with this kind of input-output transformation, this is really a single-cell computation, right? The input and output, there's a major transformation. And then you can actually think about this. So single spike, if you know about cortical release, synaptic release probability, it's very low. So for single action potential, if you compare with their impact to the dendritic integration, you would think single action potential, their major role is actually serve as a feedback. It's more important for single action potential to go back than going forward. This is my theory. Intuitive theory. I hope some theorists will turn that into formal mathematical equations. Okay, so single action potential serves as input. It's an input to a cell, not the output. The input is going back, integrating with another input to produce the real output. The real output is this dendritic plateau. The plateau will produce, plasticity. Yes. Yes. I want to admit, yeah. So this only apply in this condition. So all the theory has limit, right? All the theory can be wrong, but that can be applied under certain condition. Okay, so that's interesting theory, but everything is done in slides. Okay, with this kind of finding, people really use their imagination. Okay, this is really interesting, really powerful. So what kind of computations can be done in vivo? So people make a lot of hypotheses. The one is, so if you receive some kind of external sensory information based on the cortical circuit, canonical circuit, the parasomatic area receive thalamic input, right? The feed-forward input. But most of the feed-forward input do not produce conscious experience. Like if you turn your attention away, what I'm talking on here, even it's loud, it doesn't make much sense to you. Even you can totally ignore my sound. So without attention or without top-down input, you probably won't produce much. That's why single spike wouldn't produce much. And of course, there is context. If you pay attention, okay, to my talk, that can help. But by itself, it should not produce any output, of course. If you only pay attention to my talk, I don't produce any sound. Can you hear anything? No, right? So your attention will not produce output, of course. But if they combine together, you will generate bursting output. That's very significant. And then you will hear everything very clearly, loud and clear, right? Okay, so this is the theory. Maybe during behavior, there's information integration. Whether we can test this. Yeah, of course, I will then show something. Yeah. Yeah, so it's unknown. So here, essentially, this hypothesis does not tell about this. Although in the cellular size recording, they show coincidence detection, but here, in theory, it can be like a persistent fluctuation or persistent depolarization of the distal dendrites, some kind of slower, depolarization. Yeah. I'm not sure I understand the question correctly. So, if I may guess, you mean whether the axon projecting to different area, it can, the different type of signal can have different impact. Yeah, but it's totally unknown. I think that's potentially possible. That's why it's open question. So there are a lot of open questions. So one single cell they project to multiple areas. They have very long axon. So maybe different projection, different collaterals, they can be differently impacted by different type of pattern of action potentials. So for example, some axon is far away and low release probability, then single action potential can fail. Single action potential propagate to that location may never produce an output. But if some collateral are very nearby and have larger snaps, a higher release probability, maybe single action potential can produce impact, can cause transmission. So that's possible. The axonal diversity can be one factor how different type of action potential pattern can have different effect. Yeah, that's possible, but still it's just purely speculation. Yes. Yeah, it's a very vague hypothesis. It's not it's not quantitative. It's highly intuitive. But to demonstrate this intuition, I give you a lot of examples for fun. Okay, so this is a sensory input. If you first time saw this, it can be quite random, quite noisy, not much information. Okay, so that's a then I'll give you something for you to remember or produce some kind of impression in your brain. So that can serve as a top-down input later. Okay, so now you can probably, I think, if you have a normal vision, you will combine the top-down input with this bottom-up sensory input. That make a lot of sense to you. Now there's a person, a figure inside the image. So remember, you are receiving the exactly same sensory input, but you have different top-down input. So previous one, you only have bottom-up sensory input, so you cannot get much information. But now you have something in your brain that can serve as a internal, prediction or top-down attention, whatever you're curious about. There's something in your brain now. So that's what the internal signal means. This is one type, but we cannot. It's very hard to measure it. Okay, we only know there is something in your brain now. How can we measure it? how can we quantify it? So that's the major challenge. And there's this McGurk effect. Okay, let's see.
Ba, ba, ba,
If you
ba,
watch,
ba, ba,
okay,
ba,
so
ba.
this sound. Next, if you pay attention to his mouth,
Ba, 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 ba.
so you hear
ba,
a different sound between the
ba,
two
ba,
videos.
ba.
Okay, but they're exactly the same sound, right? So maybe I think most of you may have heard about it, but some of you may, so that's the first time you watch this? No? Okay. Oh, yeah. But I have to tell you, even you have been seeing this, but it will still have effect. So the same sound, it's exactly the same sound. If you watch different mouth shape, you will have different auditory perception. And here, if you quickly switch your gaze to this and this, you will hear different sound.
Ba, 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 ba,
Yeah.
ba.
So this is a very strong effect. Again, the same sensory input, exactly the same bottom-up sensory input, will produce a very different perception. That has to, there is information integration, sensory input, and your visual feedback to your auditory system will produce very different perception. Okay, so that's another type of information integration. Okay.
Ba.
And this is another one. It's also very strong. So these are checkerboards. Okay. So if you watch, if you look at this square and this square, can you believe they actually are exactly the same intensity in terms of grayscale? Yeah. Okay. 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 I guess people want to see this first. Okay. Okay. I guess you agree. Okay. At least what do you see? Or for me, unless you have superpower. This is darker. This is lighter, right? Agree? Yeah. But they are the same, exactly the same. Yeah, if we remove the surroundings, they're exactly the same in terms of grayscale. What happens? Anyone can tell me what happens? You have the same sensor input. Yeah. Yes. Yeah. Shadow. Very good. Yeah. Yeah, of course, these are very different examples. The exact processing may be different, but they have common theme. They have common, you know, processing. The processing is you need integration of sensory input and your internal, either prior knowledge or previous memory or other modality input. That's anything internal will interact with your sensory input. That can produce a very significant consequence. So that's in terms of computation, there's different input, produce different output, right? So your sensory input really depends on these factors. So in theory, in the brain, there may be common mechanism, similar mechanisms, or maybe not. I don't know. Even the priming effect is not clear. Yeah, I don't think people have recorded from human brain if there's a priming, spiking goes on, right? Yeah, 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 yeah. That that figure will either trigger your memory or it's produce some kind of concept. You have you have a certain category. Category can combine with sensory input. I will talk about this actually. Produce different output. Okay, yeah. So in this case, we have very strong prior knowledge. There could be two of knowledge. One is it's a checkboard. Okay, we know it's checkboard because then it must be different. Our brain deeply believe these neighboring square are different, and then there is a shadow. Our brain deeply believe the shadow can make it darker. So then we have compensate. It's actually lighter. That's why it is the prior knowledge combined with exactly the same sensory input produce undeniable perception. It's very powerful. It's subconscious. You cannot so actually one day one interesting thought is if you can ask an AI to produce the same illusion, that AI must be very smart. That AI can have this common sense knowledge. But I guess I would doubt whether an AI would generate the same illusion as we do. If that AI can do this, that means the AI is very close to us. Yes. Yeah. Yeah, there are many studies to study this kind of mechanism. So one is there's a field of multisensory integration. So basically, this is one type. So we are kind of dominant by our vision. We are vision animals. So the brain somehow really believe what we see, and you know override our hearing. The, I don't know whether there is opposite cases. Yeah, I'm I'm sure. Yeah. Okay. So there is this probably a common theme. So this is you can call this algorithm, but it's a very crude, non-mathematical, non-formal, intuitive type of algorithm. But that captures previous diverse phenomena. You have sensory input, feed forward. You have top-down or internal feedback. Together will generate your perception and judgment, and then we'll produce motor action. Okay. Okay. Then I'll show one study. I'm not sure. Oh, there's 1 minute left. Okay. 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 There's a one my own earlier study using the mouse whisker system to investigate single cell information integration of different type of source. So so this whiskers system is very nice because you can really it's really have very nice topographic mapping for single whisker. There is a corresponding area dedicated to that whisker. So this is really this feed forward sensory input mapping. At the same time, if you do a virus injection, so this is done by Tian Yimao, to the motor cortex. So because this system is really sensory motor area, so sensory and motor coupled strongly. So you will find a very strong projection from motor cortex to layer 1 in the viral cortex. So in theory, this distal input and the somatic the somatosensation input will integrate. Okay, so whether we can see this, so we can basically train the mouse to report their perception. So a mouse is head fixed, and then we randomly deliver a pole into their whisker field, and then they need to whisking to search for the object. So whenever they touch an object, they need to lick for water. So that way we can use their licking as the reporter to tell whether they actually perceive and tactile objects. Okay, so at the same time, we can use two-photon imaging to image the distal dendrites of layer 5 pyramidal neuron in the barrel cortex. Okay, so we can use we use a high-speed video to track the whisker. We know every moment whether the whisker touch the object, and the idea is if there is a simultaneous movement, active sensing, there's a motor signal coming to the barrel cortex, coincident with sensory touch information that can in theory produce object tactile perception. So I move my sensor, I have sensor input, two information bind together will produce, okay, there is an object. So this is likely to be the perception. And we can detect with the mouse licking. Okay, so that's the actual experiment, the two-photon imaging the barrel cortex. At the same time, the lick port report their tactile perception. So the bar rise at different location, so they need to report their tactile perception. And what we found is there is really global calcium signal. So for all the branch belonging to the same cell, they go off together, and they show highly correlated events whenever the animal actively touch the object and report. Okay. Let's go quickly. So if you align the dendritic calcium signal with the pole period, so there will be touch events. Okay, and then you can also compare in the dendrites the active touch, and they report active touch evoke the calcium signal is much greater than if you just use air puff. Even though the air puff can produce a larger deflection of the whiskers, even the sensory stimulation is stronger. The signal is still much smaller. So that means the dendritic calcium signal is really integrating the active movement and the actual somato sensation, sensory input. Okay, and then you can also use two-fold imaging to image the axons from motor cortex. So that's really the first study show one can use two-photon imaging to image axon activity. So the good thing is sometimes you want to read out the neural activity far away or deep in the brain, then you can actually inject the virus and image their axon terminals. And you can see really a large calcium signals. And those signals actually encoding the slow whisker movement. Okay, so these experiments really tells at a single cell level, you can integrate internal motor efference copy signal and sensory input, generate large calcium signal, calcium spike. Okay, I think I'm running out of time. Actually, at this point, I wanted to transit from bottom up to top down, because now we have some kind of algorithm, okay, although it's not mathematical form. And then we can consider whether we can, based on this, design some kind of experiments to tell how the brain combine this information. But I think this is not easy story. I'll probably stop here and tell that in the next lecture. Okay.
Any more questions? It's a very practical. Okay.
Yeah, there was already so many questions.
We have a very nice pedagogical lecture on single neuron computation.
Yeah. There is another work also that they showed that they can also they displace right? And they show the same.
Yeah, so on the viral cortex whisker system, so one study is also from Delcona. So the final story was they activate one viral, therefore work, and then try to also evoke the licking. Although there was no actual whisker. So they actually mix the actual whisker stimulation, touch, and genetic activation of their form. They quantify how much light, how they reduce the light to a minimal amount to see what the threshold is for perceptual perception of the tactile sensation. Yeah. And before that, they also try. Yeah, they try a lot of things. They try use another whisker, very soft bending to touch and see whether for I don't remember exactly experiments. Anyways, it was very interesting.
All right, let's go for lunch.
Okay, yeah, next time.